Don, thanks very much for inviting me. It's great to be here at UMBC, and very pleased to be speaking to the Brown Club of Baltimore. Very important, as mentioned, I am a Brown alum, so it's really a great pleasure to be speaking to a group of other folks who are associated with Brown in various ways. Can you all, everybody hear me okay? And I'll look to my colleague with the video camera in the back, wants to make sure that my voice is okay, so we have a thumbs up. All right. Over the next 45 minutes or so, I'm going to describe what is sometimes called brain-computer interfaces, and we'll be describing various technologies that we hope will one day help to restore communication and mobility to people with physical disabilities. There are lots of other names for this particular technology. Sometimes brain-computer interfaces is what it's called. Neural interfaces have come up in the past. Brain-machine interfaces is what the Society for Neuroscience refers to these technologies as. This is the conference that there are 30,000 neuroscientists at this meeting in DC this week. The neuromotor prostheses are another name, and some of the other names you may have read in the newspaper and medical journals or elsewhere. And as Leslie mentioned, one of the other names for this field is sometimes bionics. But bionics, as you may know, carries some baggage with it as a term. Some may remember Steve Austin and Jamie Summers from not that long ago. And for a moment of audience participation, does anybody remember who that is?
Rin Tin Tin.
It's not Rin Tin Tin, but that's a good guess. It's not Lassie. That's right. So that was the Bionic dog. That was Maximilian, the Bionic dog, who was the pet of Jamie Summers, the Bionic Woman. This was the Bionic Woman, as many of us remember her. For those who were watching TV about a year and a half ago, the Bionic Woman returned on NBC for just a few weeks before the writers' strike, and then she disappeared. But hopefully we'll continue to see an interest in this field, whether it's in TV or elsewhere. More importantly, what we're really going to be talking about, though, is how is it that we turn the ability or the desire to move into actual movement, or how do we turn a thought into action? And the pictures are various experts in turning thought into action, whether it's in the realm of dance or sports or music or art. In the lower right-hand corner, I put Curt Schilling of the Boston Red Sox, who I guess I have to tread lightly here as I talked about his contribution to various World Series. But as importantly, if not more importantly, he and his wife's foundation have given several $1 million in support of ALS research over the past few years, which is very important. It is also important, I'm going to tell you a little bit about an ongoing FDA-permitted investigational device exemption trial, so important that I mention my financial disclosures. So maybe there's somebody in the audience who will help me change that, but that's the current status. It is relevant that I mention that one of the technologies that I'll be describing, if not the main one, was built and research was sponsored by Cyberkinetics Neurotechnology Systems. That's actually a startup that was a spin-off from Brown several years ago, and they've supported some of this research. Also very pleased to report that our research has been funded generously from the Department of Veterans Affairs, from NIH, and from the Doris Duke and Dean Institutes. The most important disclosure, though, by far, is the great group of people that I get to work with every day. At the center of all of this research is Professor of Neuroscience at Brown, John Donahue, who's also the director of the now Institute for Brain Sciences at Brown, colleagues in computer science, colleagues in engineering and neurosurgery. And this is just a few of the people who sit around the table every week to think about our research and to help to move it forward. It's really, truly a multidisciplinary research endeavor, and that's one of the great things about it. Let me start off with a clinical vignette. I should have mentioned at the beginning, I spent some of my time as an acute stroke and neurocritical care physician, helping people who've either had acute strokes or life-threatening infections or injury to the brain and the spinal cord. And I know that there are some neurologists in the audience. For everybody else, this is an MRI of the brain taken back about 13, 14 years ago now. And what you're looking at are slices through, images of slices through somebody's brain going from the bottom up to the top. And so at the bottom, which is at the top of the screen, you see the cerebellum, which is in the back and the bottom of the brain. And as you move up higher and higher, you get different pictures of the brain. This was an MRI of a woman who was 42 back 13, 14 years ago, who had no significant past medical history. That is, she was perfectly healthy, when suddenly she couldn't move the left side of her body, and then she couldn't move the right side of her body, and then she couldn't speak. And when we look at this picture, this is a type of MRI called a diffusion-weighted image, and what this shows us are the parts of the brain that have had a stroke. And in simple summary, in this slide, gray is good and white is bad. And most of what we see up there is gray, except you may notice on the left in the middle, right in the center, there's a big bright spot, which if we magnify a little bit, right in the middle of the brain, that's the brain stem, specifically the pons, which sits in the middle of the brain stem. And that has had, for that part of the brain, a very large stroke, essentially disconnecting the perfectly functioning top part of her brain from everything below it. A few days later, she was awake and alert, understanding everything that was being said to her, but she was unable to move and unable to speak. And it's for people like her and for other people with similar or perhaps not as severe disabilities that we're all hoping that we can develop technologies that will restore the ability to turn thought back into action. And we'll come back and talk about her in a little while. So now let me jump ahead. Let me introduce you to a gentleman who you see sitting in the chair. He's going to control that television set, which has Dr. Phil on it, using what's essentially a remote control up on that computer monitor. So you'll see a cursor that's moving. There's some channel buttons on the left and volume buttons on the right, and he'll be getting some cues as how to move that cursor.
You want to do power? Power. All right.
So at a first pass, it may not be surprising that there's a 25-year-old gentleman who's controlling his television set. But about 4 years before this video was taken, he was breaking up a fight after a football game, was stabbed in the back of the neck, and had a C4 Asia A spinal cord injury. That is, he was unable to move his arms or his legs after that injury. But he was controlling that cursor on the computer screen, and there's a hint for how he was doing so, which is that little gray box sticking up from the top of his head. He was moving that cursor by thinking about the movement of his own paralyzed hand and controlling that cursor to some extent as though it was a computer mouse that he might have been using beforehand with his hand. So now let me take several steps back and describe how we got to that step and where we've been since then. So the discussion today is about neural interface systems or brain-computer interfaces or whichever other moniker we wish to use. And all of these systems, as defined, permit the direct, real-time, cerebral neural control over an external device. That is, taking a signal out of the brain somehow and trying to do something useful with it for someone who's lost the ability to move or to communicate. And the goal of this particular technology, if not the goal of all of neurorehabilitation, is to restore the communication, mobility, and independence to people with severe paralysis. And it's really for these goals that the great group of folks at Brown and our associated institutions that are working together in this research are really focused. So why do we need these devices? Why brain-computer interfaces or any of these neural interfaces? Well, there are many diseases that can leave somebody with an significantly impaired mobility, but still awake and alert and wishing to fully interact as they had before. And some of those are listed on the left: stroke, ALS, which is amyotrophic lateral sclerosis, or Lou Gehrig's disease. Locked-in syndrome, the state of that woman whose MRI I described a moment ago, is sometimes referred to as being locked in, that is fully aware of everything going on around her, but unable to communicate with the outside and unable to move. Spinal cord injury, multiple sclerosis, and the others that are listed there, all situations, injuries, or diseases that the ability to control one's environment is impaired as a result of the injury or disease. Conservatively estimated, there are hundreds of thousands of people worldwide with these diseases or disorders, and the assistive technology that we have available today is really quite limited. In cartoon form, the desire to move, as we all know here, begins in the brain, and that thought process, that intention to move, finds its way through a pathway somewhat more complicated than the one listed there. From the top of the brain, from the cortex down into the brain stem and the spinal cord, usually to engage movement. But for any number of these reasons, the movement may become impaired, any of the reasons listed on the left. And our goal, while there's lots of research very well focused on trying to repair that injury represented by the red X, our goal is to try to create a new pathway to take that intention to move and create a new pathway for it to enable movement and to re-enable that ability to turn thought back into action. So how do we design these systems? When we're sitting in the lab years ago, or as we do every week, to think about what we want these systems to look like in the future, there are three main components. And the one that I'll describe today is one of several that are being developed around the country and around the world right now. The three main components are a sensor, a decoder, and an assistive technology. And I'll describe those in a little bit more detail. I'm a neurologist, so this is my iconic representation of a person. What we know is that somewhere in that brain is the desire or the intention to move. And it may start there, it may start somewhere else, but we need to extract what is grossly overstated here as thought. More accurately, more conservatively, what we want to know is how is it that somebody's intending to move their hand, for example. And there are some questions that we need to ask ourselves and then answer right at the beginning. And those questions are listed here. The first is, what signal do we want to extract from the brain to enable this ability to turn thought back into action? There are lots of signals in the brain. Some are electric, some are kind of related to the electricity that can be picked up with other magnetic types of sensors. Some are related to blood flow, some are related to metabolism. And all of these signals may have some relevance depending on how we study them and how we record them, but we have to choose one of them and stick with it as we develop our technology. We also have to think about what area of the brain or areas of the brain we're interested in recording from. If we look at the monkey, which is a common animal that we've learned quite a bit of what we know about how people move from studying non-human primates, there are at least 12 different areas of the brain that are intimately involved in the control of movement. And probably in humans, there are at least that many, if not more. So we have to decide whether we want to record from one of them or from lots of them. And once we answer those two questions, we should decide what sensor we want to use. If we're interested in recording from the whole brain, we may use an MRI, a big magnet that you're probably familiar with. Not very portable, but certainly is something that can get lots of brain activity recorded from. Or in our case, we may want to record from just a handful of neurons inside the brain. And that's what I'll be telling you more about. Once we've made those decisions, then we need to take all that information that we're recording and put it into what I guess most respectively describe as a black box. This is the decoder. This is the software or perhaps hardware that takes whatever signal we've recorded from the brain and helps us turn it into something useful. And for today, describe that the neurons, the brain cells that are involved particularly in the control of movement, that's what we're recording. So we want to look at what's listed up there as a firing rate vector, but really we want to study the firing patterns of those neurons in the brain and convert them into the hand motion that somebody with paralysis wishes that they were making. Between that signal, the firing rate vector, and the hand motion is what I'll refer to as either a filter or a decoder. It's the software that takes that signal and converts it into the intended movement. So those are two of the three components. Before we're done, we do have to think again about what it is that we want to do with all the information we're collecting from somebody's brain, and what it is, more importantly, that they want to do with that information. For the woman who I described, who's unable to move and unable to speak, if we could restore the ability to type or to browse the web for entertainment or education, to use a text-to-speech application, all those would be extremely helpful in improving her quality of life. For somebody like the gentleman whose video you saw, who is able to speak, and you'll see him speaking in just a little while, but who's paralyzed, who's tetraplegic, that is unable to move his arms or his legs, if we could improve his ability to control a powered wheelchair, maybe to control semi-autonomous robots to help with his activities of daily living, all of those would be helpful. For people who have lost limbs, whether the result of trauma or vascular disease, there are some really superb prosthetic limbs that are now being developed. But what we don't yet have is a great way to control those limbs. And if we could take this signal out of the brain, the desire to move, and convert that into the control of a very advanced prosthetic limb, then maybe we could really allow somebody to get the great control that's available by these limbs, but isn't truly available because there's no controller for it yet. The dream for the research, though, is listed there at the bottom, at least for people with spinal cord injury or brainstem stroke, which is to one day reconnect brain to limb, to allow somebody to think about moving their own paralyzed hand again and for that hand to move. And that might be done by taking these brain or movement-related signals from the brain and connecting it to other technologies that are implanted in the limb that enable movement, that stimulate the nerves and muscles. And I'll come back and describe a little bit of that research in a little while. But that's what we sit about. We think about that diagram regularly and some of the detail that's in there. I don't know how many of you have had the chance to listen to a neuron firing away, or at least to the sound of an amplified neuron. So that static is music to the ears of an electrophysiologist. That staccato sound is the sound of one neuron in the brain being recorded, as it has been for more than half a century, usually by single electrodes, as appear on the picture there, being placed into the brain, generally of animals, but more recently of humans, to record these action potentials, these waveforms. And these action potentials are the way that neurons communicate with each other, the way that neurons communicate with muscles. It's the language of the nervous system, and it's really what makes us all who we are. The change in those firing patterns, the rate of that staccato sound, and the way that this ensemble of neurons, the 100 billion neurons that we have in the human brain, those firing patterns are what we're hoping to study, or at least a small segment of those 100 billion. What we're hoping to study to understand what those rates mean, what those firing patterns mean, and to allow those firing patterns in certain parts of the brain to control an external device for somebody with paralysis. This is one of my colleagues from grad school. He and I spent a lot of time together. Much of what we've learned about, as I mentioned before, about how we move has been learned with the assistance of non-human primates in the laboratory. It turns out to be fairly simple to play a monkey, to teach a monkey to play a video game, which is what he's doing there. He's sitting comfortably in the chair, has his hand on the joystick, and he can control the cursor that's represented up there. And as he moves that cursor left and right, if he's keeping it in the center of the moving box, whenever he does that right, he gets a squirt of apple juice or whatever his favorite treat may be. And animals will be happy to do that for hours on end, so long as you've selected the right cocktail, which for him was a combination of applesauce and apple juice. And I only mention that because it took about a month or two of my graduate study to figure that out. Once that's been achieved, once the animal's playing, whether it's this game or any other, that we can watch their hand movements carefully, then people have recorded from the brain, the part of the brain that controls that hand, to correlate the movements of the hand with the activity, that is the brain activity, that's recorded at the same time. This is just a quick slide that I'll go through very briefly. It's a post-it note that summarizes, for me, about 40 years of science. It tells us all of the different variables in the firing patterns of individual neurons in a part of the brain called the motor cortex, or the primary motor cortex. This is a part of the brain that's instrumental for, in particular, the fine control of the hand. And whether it's the force that somebody may be placing when they're picking up a water bottle or squeezing on a water bottle, or the direction of movement, these are all quite important, and they're encoded to different extents in the firing pattern of these individual neurons in the brain. That up in the right is just a reminder again for me that not only are these single action potentials recordable by electrodes, but there are other signals as well that can be recorded. For the aficionados here, those are things that we call multi-unit activity or local field potentials. One neuron at a time, though, is a hard way to understand the nervous system. It takes a long time to understand what one neuron is doing, and back about beginning 15, 18 years ago, people began to record not just single neurons in the brain, but a handful of neurons at the brain at the same time. And as we did this back in the mid-90s, these small boxes each represent the activity around one electrode in the brain. And so in any of these groups, there were 24 electrodes in the brain of this happy, healthy, neurologically intact monkey playing a video game. And what can be seen even just on the left at rest, the dark colors are not a lot of activity and the bright colors are lots of activity. That when the animal's at rest, not surprisingly, in this part of the brain that controls movement, there's not much going on. It's pretty quiet. But as the animal flexes his wrist or extends his wrist to play that video game, there's a different activity. And again, probably not surprisingly, as the animal reaches up to grab a peanut out of my hand, there's a whole lot of activity in that part of the brain that controls movement. One of the reasons that's important is just at a quick glance at these 24 sites in the brain as opposed to just one, we can see fairly quickly, at least in the color-coded map, that these look very different. That maybe if we understood not this color code, but what those firing patterns meant at just a handful of sites in the brain, we could guess or predict what the animal was doing based only on the activity of a small sample of neurons in this part of the brain called the motor cortex. So with those and other findings at about the same time, it became clear that not just the single electrode, and certainly not the fine wire electrodes that we were using early on, but some other technology needed to be developed to allow us to record, not just in the laboratory, but more importantly in the future in our human patients, from lots of cells in the brain. So one of those was developed originally at the University of Utah by Richard Norman, and subsequently through John Donohue's lab at Brown and Cyberkinetics. This is what looks quite large on the screen is a 4x4 millimeter array of 100 electrodes, 96 of which are active. And altogether, they're about the size of a quarter of the nail on your little finger. So one of those arrays, very tiny, can be placed into the brain of either animals or humans, more recently humans, and we can record again from lots of places in the brain, giving us the chance to understand what all that brain activity is doing, which we'll talk more about. So how's that done? A small hole is made in the skull, which is common in any neurosurgery, actually required for any cranial neurosurgery. That array is dropped in, the little piece of bone that was taken out to make that hole is put back, and everything's sealed back into place, and the skin is put back, and we're done. Are there any neurosurgeons in the audience? I should check that nobody's going to yell at me for that brief summary of all of their training. What's important about this photo or this picture is that the array that's in the brain is connected by some fine wires up to the pedestal that you see, which is the gray plug. Currently, that sticks out up off the top of the head. And whenever we want to record, either in the laboratory or in our case in our clinical trials, from this part of the brain, we have to take a medium-sized plug and plug it in to that small plug that's up at the top. And I'll tell you a little bit about some of the work going on at Brown that'll eventually help us get rid of that plug. So we talked about this part of the brain that's important for the control of movement. How well can we predict that animal's hand motion from just these cells? Well, here's some work from, again, from John Donohue's lab back a few years ago. The yellow line that you see on the left is the animal's actual hand path, that is, as he's playing one of these video games, in this case with his hand on a tablet. And the red line represents the decoder's prediction based just from the activity coming out of the brain of what that hand path was. And it's hard to see in this light. There's lots of little dots in the box on the right that represent the activity of 24 cells simultaneously. In real time, what you see, again here in white, is the animal's actual hand path as the animal is moving around on this tablet. And the blue dot represents what the decoder thinks the animal's hand is doing based only on the activity of these handful of neurons, a couple dozen neurons in the brain. So in real time, with the help of our folks in computer science and computational neuroscience, we can predict, again, from this happy, healthy monkey who's playing a video game, just from his brain, what it is that his hand is doing. So that's an important first step. But the next step is to see whether the brain and the signals coming out of the brain can actually control the cursor directly. And the way that's done is starts out just as I described before. Have an animal trained to play a video game, record from the part of the brain that's important for movement, build one of those decoders that takes the brain activity and turns it into hand movement, and then play a small trick on the animal, which is to disconnect the joystick. Don't know how many of you have had the chance to work with monkeys and play with them in the laboratory as they're playing these video games, but when you disconnect the joystick, you always have to pause because you get a bit of a reaction, which is usually a dirty look. Because the cursor that they were controlling, or the joystick they were controlling before, suddenly stops working. And the cursor that they were controlling originally by hand doesn't quite work the same way anymore. What happened back in early 2000, 2001, when John and his colleagues in the lab and Misha Soraya, an MD PhD student at Brown, were doing this, was once the joystick was connected, the cursor was then connected directly to these brain signals. That is, the decoded brain signals that were predicting the animal's hand movement were placed directly in control of the cursor on the screen. So what's represented here in the graphs, the green dots represent the actual hand path of the animal, which started to move a little bit strangely. The red dot is the target, and the blue dot represents what we'll call a neural cursor, a cursor that is moved not by any physical activity, but by brain activity alone. And you can see on the left that the blue cursor kind of takes a little bit of a rotation and then finds its way to the target. On the right, the animal adopts a somewhat different strategy. Indeed, the animal begins to do what perhaps is what many of us often try to do, which is to accomplish as much as possible while actually doing as little as possible. And what the animal did here is take his hand and place it on the side of the chair that he was sitting in. So the green dot is perfectly stationary, but the blue dot represents this neural cursor moving from point A to point B, getting to the target and allowing the animal to get the squirt of juice. So let me show you that in real time. The green dot here represents the neural cursor. This cursor is being controlled by the animal directly through brain activity. And whenever the green dot hits the target, which is the red dot jumps to a new location and the animal gets his squirt of juice. So you may see that the green dot's a little bit wavy. I know there are a few neurologists in the audience, perhaps a little bit ataxic might be a good description. And we can come back and talk about that. But the animal achieved the tasks, and this was the first demonstration of the two-dimensional control of a cursor in a closed-loop fashion by a non-human primate. So this is cursor control directly from brain activity, and yes?
How quickly did the animal realize that it was useless to move his hand?
So sometimes the animals, and I wasn't part of the group at this point, but as both John's group and the others that are listed here, I think there's a few more listed at the bottom of the screen. Sometimes the animal will keep their hand at rest, at other times they'll continue to move it around. So whether they truly acquire, quite in the way that I described, the knowledge that their hand didn't have to move is a difficult question to really get at. But it did become clear that their hand movement was different, and in some cases they stopped moving it. And certainly feel free to raise your hand and stop me at any point. Yeah. So this was the first, as I said, demonstration of closed-loop neural control of a two-dimensional device. And what's put up at the top is this was the tongue-in-cheek referred to as the food from thought experiment. I guess every time the animal thought about moving the cursor the right way, it got a squirt of juice. This was really the proof of concept, again, for a neurologically intact, happy, healthy animal, that maybe we could take these same recordings, these same set of cells from somebody who has paralysis, record from them, understand that brain activity, and allow them to control an external device again. So from this research, the company that was spun off, Cyberkinetics, formed a pilot clinical trial and named the device the BrainGate device, or the BrainGate neural interface system. And the goal of the ongoing pilot clinical trial, or a goal, is to determine whether somebody with tetraplegia, unable to move their arms or their legs, might be able to control a cursor on a screen just by thinking of the movement of their own paralyzed hand. So if this works, the same array that was built through so many years of federally funded research is placed into the motor cortex, again, that's part of the brain, that's very important for the control of movement. That array is then placed, the plug is sticking out of the top of the head, and on any of the recording days of our clinical trial, that yellow plug up there, as it's shown, is placed into the plug that's protruding from the top of the head. All that neural activity is sent down to some amplifiers, and if it works, when somebody imagines moving their hand, the cursor on the screen will move in much the same way. So our trial's been ongoing for about 4 years now. The people who are enrolling in this trial have either spinal cord injury, brainstem stroke, muscular dystrophy, or ALS. Some of the other inclusion criteria are listed there. Once the surgery is done, and the original surgeries for this trial were performed by Gerhard Fries, who is a neurosurgeon at Rhode Island Hospital, who moved just a few months ago. Once the device is placed, the participants return home or to wherever they were living before, and the rest of our clinical trial occurs right in the place that we hope this technology will eventually be helpful, which is where our patients and our study participants live. So here's some photos from that first implant, which was placed back in 2004. The gentleman in the first video who you saw, you'll see a few more in a moment, was the first to receive this device. There's Gerhard's the tall one in the picture, and standing next to him to Gerhard's right is John Donahue. The original procedure took just about 3 hours. It's been shortened to around 2 hours in the subsequent surgeries. And those are some CAT scans, and you can see this little device that sticks up from the top of the head when the surgery is done. One may wonder where exactly we're placing this device. On the left are some images from a paper a little over 10 years ago pointing out that there is anatomically a part of the brain that's fairly easy to find. That's the central sulcus, which is the dark line that separates the frontal lobe from the parietal lobe of the brain. And right in front of that, on functional MRI, if people who are not disabled, people who are moving their hands, do so in a functional MRI study, that part of the brain will light up. That is, we know that that part of the brain is involved in the control of the arm and the hand. And thankfully, our first participant's MRI, which is over on the right, looked just like it did in the textbook. And so we placed the array, again, right into this part of the brain, very important for the control of the arm and the hand. Mention that there have been 4 participants in our trial so far. Through today, there have been 2 people with spinal cord injury, one with a brainstem stroke, one with ALS. Highlight the third person, who's the woman who is still in our trial. I'll tell you a little bit more about her momentarily, but she's been in our trial now for well over 2 years, and I think we're at 2.9 years, nearly 3. And I'll tell you more about her in just a second. Whenever somebody's describing an ongoing clinical trial, important to mention the adverse events and the serious adverse events that have occurred. Those are listed there. The only 2 that are related directly to the trial itself have been the development of a postoperative fever for 24 hours and 1 participant related to the surgery, not to the surgery itself, but to being on a breathing machine and the general anesthesia. And the other study-related adverse event was the expulsion of 1 vicral suture that normally dissolves, but when it doesn't dissolve, the skin does what it's supposed to and gets rid of it and expels it. However, this is a population, because of their tetraplegia, who's at risk for infections and for other adverse events, and those are listed here as well. I'll come back and describe a few of these folks as well, in particular the gentleman who died during the trial, not a result of the trial, but certainly an important event, which I'll describe. So one of the questions that we had to ask ourselves, if somebody hasn't used their hand in 3 years or 9 years, what is this part of the brain going to do that used to control the hand? What is it doing now? Has it taken over some other function? Is there some other function that is now being used or being helped by this part of the brain? Or is it still trying to control the hand? So what's listed on the screen are from our first participant. This is the gentleman with the spinal cord injury, imagining a series of movements, most of which he cannot perform, while we record, at least on the left, from 2 cells simultaneously. And each of those little ticks up at the top represents 1 action potential, and the line below it represents just the averaged activity. And what we see is he's imagining either opening and closing his hand, or rotating his wrist and his elbow, or opening or clapping, moving his shoulder, moving his knee. During these activities, we wondered, what is this cell, or what are these cells going to try to do? And if we look at the top of the screen, while you imagine the first of these, which was hand open and closed and wrist flex extend, that neuron was kind of firing away at its original baseline activity. Wasn't terribly interested in what he was trying to do. But as soon as he imagined opening and closing his hand or trying to clap, that neuron started firing a whole lot faster. And when he started imagining or attempting these other activities, the neuron went back to its original baseline firing rate. And below it and in the other graphs are some other examples of neurons that have a very different activity pattern. So here, without any learning, without any special instructions, the moment that he tried to do something with his hand, that neuron started firing away, probably just the way that it did before he had a spinal cord injury. And it was when we saw graphs like this and we listened and we heard that activity on the oscilloscopes and the amplifiers, that's really what told us that we would be able to take that activity, those cells that are still trying to do what they used to do, and to convert that activity to the control of an external device, such as a computer cursor. So The performance of our first and, to a smaller extent, second participant in the study were reported a little over 2 years ago now. There is sitting, as you can see in the chair, that's Abe Kaplan behind him, who's one of the technicians in the study, who's plugging him in, that is plugging this, this pedestal into the plug that brings the neural signals back to the computers. And what I'd like to do is show you a few videos of this gentleman controlling a cursor just by imagining the movement of his own hand. You'll hear him talking a little bit. His voice will be a little bit raspy. He's on a ventilator as he's doing it, but he's going to describe to some extent what it is that he's doing.
Okay, so here's the cybernetics desktop. What are you going to do first?
I'm gonna open my email first.
Okay,
So this is a simulated email program.
you're going to open the first one.
And it says, congratulations, you are doing a great job.
And that cursor that you see is what we call the neural cursor. He's moving that cursor by imagining or attempting to move his own hand.
It says, hi, we'll talk soon.
Great. Now can you exit back to the cybernetics
I
desktop?
want to exit.
You can exit. Excellent.
So because of his injury, he hadn't been able to draw in a few years.
Draw
So
a circle. Wow.
we brought back Mac Paint, for those that may remember that program. This was an eraser at the top and an inkwell down at the bottom. And so that's a first attempt to draw a circle.
And I
Okay.
erased it.
All right, let's try it again.
Here we go.
So that's not a circle either.
You avoided the eraser that
But
time.
as Abe mentions, he saw the obstacle and he avoided it. We asked him to do some other things for about 8 minutes, and then we asked him to try this again.
To draw a circle.
All right. Excellent. Now let's try going back.
And he goes out of his way to complete the circle just to prove to us that, that he can really do it. I'll show you some other examples of control of virtual or computer-based devices, but one of our hopes is that we can get control over physical devices, and particularly prosthetic limbs for people who've lost their limbs, as I mentioned, due to either trauma or vascular disease. One of the reasons, one of the goals of our Department of Veterans Affairs support is to see if we can help develop a technology that will allow better brain control over the great prosthetic limbs that are now being developed. So you can see just a little bit, you can see the hands just on the left of this lower screen of our participant in the trial. What he's looking at is a real prosthetic hand that's used for people with for distal amputation. That hand is facing him, and you're going to hear him say open and close just so that we know what it is that he's thinking or trying to do. And depending on how the audio is in here, you may hear his initial response to his ability to do this. Looked like his eyes were closed there. He's actually looking down at the hand. So used to play video games prior to the injury. He wanted to restore the ability to do that. So in a very retro demonstration, we brought back Pong. This, I guess, is neural Pong. So he has neural control over that cursor down, or the paddle down at the bottom.
You got it? I haven't announced it yet.
Really?
The two voices you hear in the background are both Brown grads, Abe Kaplan and Misha Soraya. It's kind of hard not to root for him as he's doing this. Yeah. It's just, again, another video game. The goal here is to capture the targets and avoid the obstacles.
Go ahead. That's a great one.
That voice was Miriam Soleil, another Brown undergraduate who's involved in the work.
Good.
Damn it.
Go ahead.
So my feeling is that that particular video showed some of the precision that he had. I actually think that harder task was better than some of the earlier videos, which were somewhat easier. And we can talk about what the relevance of that may be for our recording from this particular part of the brain, if anybody's interested. That's Misha Soraya again. He's going to place what's a piece of candy in kind of a B-grade movie near this robotic arm. This was the one and only time that this robotic arm was introduced to our clinical trial participant. That was done about 20 minutes before this video was taken. Our participant's job is to grab the piece of candy, again under neural control, and then to move the candy back towards Misha's hand and to drop the piece of candy back into it. So there, under neural control, he's grasped the candy. He's now going to rotate about what I guess we could argue is the shoulder of that robotic arm, extend at the elbow, and then release the candy to drop into Misha's hand. So rudimentary for sure, but very quickly our participant was able to take this link from the brain and get real control over a real physical device. Let me change gears just a little bit and talk about ALS or Lou Gehrig's disease, another disorder for which we hope this and other technologies will be helpful. ALS is a neurodegenerative disease that some of you may be familiar with. It causes not only injury and death of neurons in the spinal cord, which is what leads to the progressive paralysis, but also death of neurons in the motor cortex, in this part of the brain that at the moment we're recording from. So from a science standpoint, we have some important questions to ask. The first is whether in somebody with advanced ALS we can record anything usefully in this part of the brain. Even if we can, does that part of the brain change its firing activity when somebody tries to move? And even if that's true, is it possible that this part of the brain can be harnessed for control of an external device? So our first participant in the study was about 37 when I met him. He had been diagnosed with ALS 7 years beforehand. That was back in 2006, 2005 when I met him. He received the same brain gate array placed into the motor cortex. And when we met, he was tetraplegic, that is, unable to move his arms or his legs, unable to speak, was on a ventilator. He had an occasional flicker of 2 fingers of his hand, and he was able to turn his head left and right. And it was with that head turning and with a button that was placed on the headrest of his wheelchair, which he was able to control, that he was able to control an assistive technology that allowed him to type on a computer screen. And with that technology called Easy Keys, he demonstrated repeatedly that he was fully awake and alert, had great insight into our research and a great sense of humor, and kind of demonstrated both of those repeatedly and guiding us what he felt about the research as we were going along. Tragically, about 10 months into the research, he died as the result of an inadvertent disconnection of his ventilator at night. And clearly tragic for his family, as well as for us on the research team. What it really brought home to us was reinforcing the fact that even today there is no technology that provides 24/7 ability to communicate or to ask for help for somebody who is in this nearly completely locked-in state, awake and alert but unable to communicate. And I'm hopeful that this and other technologies will be developed quickly that'll restore that ability and more. He continues to inspire our work, and I'll tell you just a little bit about him and his performance and help during the study. So one of the questions that I asked just a moment ago is, are there any neurons that are working in this part of the brain? That, again, I realize is, well, turn that off, maybe, is annoying to most people in the audience. To me, it's a great sound, the sound of neural activity in the background, and all of those little pink and blue and I think maybe yellow somewhere, little squiggles are individual action potentials from one or maybe a couple neurons that we're recording from across this array. So there's lots of activity in the motor cortex of this particular individual, n equals 1, with advanced ALS. So that was an important first step. Another question that was sometimes asked is, how long does it take for somebody to control an external device just by thinking about it? How long does it take to learn this particular technique? So here's the very first day that our participant with ALS ever tried to get control of the cursor. You see, there's a gray jacket, that's his chest, his head's up above it, there's Abe again. Abe is going to describe to him on this first time exactly what it is that he's supposed to do, which is to control the tiny white cursor that you'll see up on the screen and to move it on top of the orange square.
Basically, you see a grid of squares, they're all blue. There's 16 of them on the screen. And you'll see a hand, which is controlled.
There's the very tiny white cursor that you may see.
Basically, the goal is to move the hand to acquire the orange square as soon as it turns orange.
So before Abe is done providing his instructions, our participant has already gained some control over that cursor and is able to move it from its location over to its target. Again, a little bit wobbly, and we'll come back and I'll show you some improvement on that more recently. On another day, or actually on most days of the trial, either our engineer at the trial, our technician, or the computer or myself asks our participant to try to do something. On this particular day, our participant's 5-year-old son sauntered into the room and kind of took over. So on this day, you'll see that the filter was quite good at allowing the cursor to move up, down, and to the right, but it didn't really let the neural cursor move to the left. So the voices you'll hear, the young one is our participant's son. The woman's voice you'll hear is our participant's mother or the son's grandmother.
Want to see something?
What?
Want to see something? Watch out for this, okay? So you can tell your dad to move either up or to the right. So this is up, this is the right, or this is down, okay?
Okay. Down. Move down. Move up. Move to the side. Move right. Move left. Move left.
Left is the hard one, so give him a second. It's okay, I'll try it. Keep practicing.
Alright, now tell him down.
Down.
You know how he's doing that? He's thinking about it. Your daddy is amazing.
I agree. Our participant in the study had asked us several times if he'd be able to play some video games with his son. So this was a nice moment for all of us on the research team, as were some other examples over the trial. It also taught us a very important lesson, which is you really have to make sure everything's working right before you let the 5-year-old into the room. And certainly don't let the video record it otherwise. But that was a nice moment. Let me bring you back to the first MRI that I showed you. This now, we're going to advance about 9 years. This is the MRI of that same woman who had the brainstem stroke, who I mentioned was suddenly unable to move and unable to speak. What you may recognize is we're kind of taking a look at the side of the head now with the nose up in the front. Most of the brain looks the way that it's supposed to, but right down in the middle, and there's my cursor, this part of the brain, the brainstem, is supposed to all be gray. And what you see is a large black hole right in the middle of it. This is the stroke that's nearly completely transecting the pons, separating the perfectly functioning part of her brain that allows her to think from everything below it, which allows her to move and to speak. When she was recruited to the trial, she had regained a movement of her head, able to move her mouth, but still not able to speak, and no functional movement of her hands. This is a sample of some of the neurons that we're recording from her motor cortex as she imagines opening and closing her hand, which is something that she can't really do.
Close your hand. Relax. Open your hand.
That's the sound of one neuron that's firing away the moment that she tries to open that hand.
Relax.
And as soon as she changes what she's doing or relaxes or closes it, that sound, that firing goes away from that neuron.
Relax. Open your hand.
Now, not all neurons are that pretty, or that cooperative, I guess. But it's an example, if we can take neurons like that, or even some that are less friendly, and convert that to the control of an external device, it should be fairly simple for somebody to get that neural control that we desire. So we've improved, with the help of our colleagues in neuroscience and computer science at Brown, the control over this cursor. We're now using a different type of filter, this being a Kalman-based filter. I know there's some engineers in the audience. And she's got neural control over that cursor that not only allows her to move to the lit-up target, but also to stop on it and to click. And for her, the click is the imagined squeezing of a hand, not that different than somebody's without a disability if their hand was on a mouse, just clicking the buttons of that mouse. So she just has to breeze through the center here. And then she'll find her way back up to that lit-up radial target. And when she gets there, she'll stop on it and click. And turning it from that salmon color to red. Here's just another example of the improved cursor control that we have recently. Now she has neural control over that black dot. This is a somewhat different task. The goal is to get it on top of the red dot and again to stop and to click, and whenever that happens, the red dot jumps to a new location. So for me, it's these demonstrations of point and click that were really some of the first times that I felt that we were on our way to developing an assistive technology that would be helpful for somebody with paralysis. As one of the neurologists involved in the trial, it's my job to make sure our participants are doing okay during their participation. So every month I have to ask them what's called a mini mental status examination. What day is it? What time is it? Where are we? There's a few other simple questions, including what season is it? And back a few Julys ago, I asked our participant to answer these questions for me, not independently, but while she was connected to this BrainGate device. So here she has neural control over that cursor. When she gets over to the letter of interest, she'll stop at it, and she'll, again, imagine squeezing her hand, which is the click equivalent. And slowly but surely, she'll tell me what season it is. And she eventually does that without error. It's slow, without a doubt, but when I asked her to compare this to the technology she usually used, which is either a translucent eye board or an eye gaze-based system, or the same head button that I described for the other gentleman, she described this as being much more natural. Now, maybe she was saying that because I was the one that was asking, and she didn't want to make me feel bad, but my suspicion is that we're somewhat on the right track of developing a useful technology for people with paralysis. The other thing that she's been asking me to do for a few years now, nearly 3 years, is to control her own powered wheelchair under neural control. Haven't quite yet been ready to do that for regulatory reasons, if none others. But here we did the next best thing, which is to allow her to control somebody else's wheelchair. So you see her sitting in the chair. She's thinking about the movement of her own hand as though she was controlling the joystick of a powered wheelchair. We'll zoom in on that technology in just a moment, called Roll Talk, which is a nice assistive technology that comes out of Norway, very easily programmable, and we can work with that very nicely. And her job is to get that wheelchair over towards the door, in between the two chairs, the two obstacles, basically, that are in her way. So we'll zoom in, see 4 buttons, up, down, right, left, and then stop. Every time she stops the chair, it rolls back just a little bit. Get a quick view of her hand, which is in this chronic upward or supinated position.
Go right up to it and then stop.
There's John Donahue's voice in the background.
Fantastic, very good.
So she was happy, the research team is happy. As I mentioned a few times, there's a device that's sticking up from the top of the head. That's something that has to go away. It's not a medical technology of the future. There are not going to be hundreds of thousands of people with wires or little devices protruding from the tops of their heads. And I say that initially with great confidence, and then I think about how many people, myself included, have 2 little white wires draped from our ears in various circumstances, hopefully not while driving, but occasionally we've seen that too. This needs to become a fully implanted medical technology, just like deep brain stimulators or cardiac pacemakers developed. Both of these were externalized initially, had a percutaneous component. So we're beginning to work on just that, and I'll show you that a little bit more in the next slide. What we want to do here is take that fully implanted component and allow somebody to control their own limb again. Take the brain recordings, the intended movement of the hand, and control movement of the limb. So together with our colleagues at Case Western and the Cleveland Functional Electrical Stimulation Center, which really a great collaboration, we're beginning to make progress towards that goal. The folks at Case are experts in this FES device, which has now been implanted itself into to several hundred people, I think 300 or more. Was mentioning getting this device fully implanted. Archer Nemiko, professor of neuroscience at Brown, has made great progress in taking all of this cart of amplifiers and computers that sits behind our participant, shrinking almost all of it down to chip scale, and that's one of the models of it that you see there. Versions of that have now been implanted, not in humans, but in some of the research animals, beginning to get some of these recordings and proving, in fact, that we're on our way towards a fully implanted technology. Those recordings would then be sent out either through infrared or through fiber optics to other parts of the body. Just to finish up, most of what I've been describing has been in the realm of neurorehabilitation, trying to restore communication and mobility to people who've lost that due to disease or injury. There are other potential uses for this technology of sensing, of understanding what this brain activity, what this neural activity really means. Again, I know there's some folks in neurology and neuroscience here. Deep brain stimulators for Parkinson's disease, that is a very long implanted technology that's been around for a while now. When those devices are placed for people with Parkinson's disease or other movement disorders, they're turned on and everybody goes home to some extent. That device is always on. Maybe it would be better if that device wasn't always on, it was only on when the person needed it to be on. That might help it to work a little bit better, and even if it didn't, it might help to save the battery, which currently needs to be replaced every 7 to 10 years. Probably more importantly, more likely for this particular technology, is the use of it to understand epilepsy and the development of seizures. Seizures, as you may know, are the unusual, unexpected, and usually vociferous electrical activity of the brain that results in an impairment in consciousness and sometimes of significant unintended movements as well. That results from individual neurons beginning to fire in patterns that they're not supposed to. And this is really the first opportunity that we've ever had to study in patients who are already having surgery for other reasons with epilepsy, that is to try to reduce their seizures, what these ensembles of neurons mean, what these individual brain cells are telling us, and whether we can begin to understand how seizures develop and maybe predict when a seizure is going to occur. And if we can predict them, maybe we can suppress them. And we're beginning to do some of that work as well. In the neurocritical care realm, as I mentioned, I spent some of my time as a neurocritical care doc, where patients with severe injury or disease of the neurologic variety are taken care of in the neuro-ICU. We have lots of tools available to us right now to study different activities, different types of activity in the brain, pressures and temperatures and pH and oxygen content. None of these things are really what we care about. What we care about is whether the brain cells are working or whether they aren't. And if they're not, there are things that we can do about it to try to save those brain cells as a result of the evolving injury that can sometimes happen, whether it's traumatic or due to disease. So maybe with this recording technology, we can begin to understand better and understand in advance when somebody with a severe brain injury is becoming more ill, and that can get us to act a little bit more quickly and more accurately to save that part of the brain that may be at risk. So with that summary, just pause to thank again the great group of people who I've had the opportunity to work with. It's at Brown. This is really where this activity, together with my colleagues at Mass General, is focused. There are folks from the VA, folks from the company that have been very helpful along the way in helping us to do this. And I hope that the research will continue, and that I can come back in a few years and show you some additional advances. And thank you very much for your time.